all come.
That is true, yes.
Come by different groups, and it's all an invention.
Okay, I need to save your presentation for the postdoc, so this in there. Oh, there's only certain ones you want saved. Okay. Do you want to just email to me? Just it should be
That's one of the CEOs, like millions and millions of dollars because the amount of money to bring back.
I was going, that's not all my stuff.
Let me just put that first on probably because I have also a jump drive. Did you leave that in?
I know, we'll bring
You're going to go through the alphabet, huh? A, B, C, D?
Yeah.
Tired.
But,
It's 10 hours.
And then I can just.
So we got back at like 7 yesterday. Unpacked everything, and then this morning, like, went grocery shopping. Did all that.
So yours is this one?
Yeah, that's me.
I just gotta find a new job. This one. You guys went to Florida? Yeah. Florida?
Oh, okay, yeah. Yeah, when I send them to the postdocs, I usually send them through file transfer because they're all big. Right.
Okay.
We're recording.
Okay, so I'm really pleased to be able to provide the introduction for Dr. Heike Muenzberg from Germany. She's an associate professor here at Pennington, but I wanted to tell you a little bit about how she got here. She has her education from institutions in Germany, I guess a Master of Arts in Biology, and then another degree in biology at another institution there, or a PhD there, I think.
A PhD in Hamburg, yes.
In Hamburg, okay. And she was a postdoctoral fellow at Harvard Medical School, so she's been at another institution in the U.S. before she has come here to Pennington. And if I'm reading this correctly, before you came here, you went back to the University of Michigan.
Exactly.
Where she was a research investigator and then a research assistant professor there. And she arrived here at Pennington in 2008. And so currently here at Pennington, she is an associate professor. With that brief introduction, I'm going to turn it over to you to tell us about your work.
Thank you. All right, so I'm going to try to, let me just, this is, I think we have to do it afterwards. Let's say, okay. So it's going to be a little bit different than some of the other talks that you have seen because I'm going to emphasize today a little bit thermoregulation and try to make a case how thermoregulation is also important for body weight regulation, which is not always put in the same corner. And I mean, certainly we can agree that this one here is very familiar to people in Louisiana. We have to thermoregulate a lot to stay cool. And if we are more in the north, we have more trouble with staying warm. And so overall, I would like to emphasize initially that if we talk about physiology and the physiology of humans or any organism, we are talking about adaptations to environmental changes. And so those changes are typically involving all of the organs, the brain, obviously all the organs that we have, and they have to work in harmony to help us to adapt to the environmental changes. And so most of the presentations you're going to see will be about the situation, and obviously we are interested to cure human obesity, but I kind of have this little dog here, and you see it also in cats. We have the same problem of obesity. But if you think about physiological adaptation, also the other part of that token is important. So also in lean nets, it's the same neuronal circuits, it's the same physiological adaptation pathways that are important for both situations, either for regulating an environment with a lot of food or an environment with very little food available. And then today I'm gonna talk about two other important adaptations to cold environment and to a hot environment, and also as humans we have to adapt to it. Even though we can mostly stay at thermoneutral conditions, we still have situations where we have to be able to cope with the cold, and we can usually amazingly do that. And all of this to regulate body weight and to regulate body temperature can be put together in that word homeostasis. So the two names that you have to keep in mind for homeostasis are really these two gentlemen, and Claude Bernard has been a French physiologist, and he really introduced the concept of homeostasis. He did not use the term, but the concept overall, that was really introduced by him. And he realized first that the stability of the internal environment, which he called the milieu interior, builds the basis for free and independent life. Do you know what that was, this milieu interior that he was investigating? Someone know that? What part of the body did he look into? It's the blood that he was looking at. And so he worked actually on blood glucose homeostasis, and he demonstrated basically the first classical feedback system, and that was about the liver function and maintaining blood glucose homeostasis, where he showed that the liver is really important to provide glucose when the blood glucose level drops, or to take out glucose and store it if there's too much. And then Walter Kinnan, he was the one who really termed the word homeostasis, and he also termed steady state that we use a lot of times when we talk about homeostasis. And also Walter Kinnan, he described main features of homeostasis, and that is constancy in an open system, which is our bodies, requires mechanisms to maintain constancy. And also homeostasis is a result of an organized self-regulation, so we do not have to memorize to keep our body glucose, blood glucose constant, it just happens. But something needs to organize it in our body, obviously. And so if you speed now forward several hundred years, so this is the modern way how we look at homeostasis. So this is a homeostatic control loop, which classically acts in this negative feedback way. And in this situation, I kind of wanted to talk about fasting. So if the body is in homeostasis, and then the system is being insulted, there's a stimulus that flips it out of homeostasis. So if that would be fasting, then you have an imbalance that appears. And so this imbalance produces changes in certain variables. So does anyone know what, if you're fasting, what is the typical thing that will happen that would change? Any ideas? Priscilla, what would happen? What is
levels.
Hmm?
So fasting, blood glucose levels.
Blood glucose level?
Temperature.
And what happens with the body weight?
Goes down.
Goes down. What goes down particularly if the body weight goes down? Adipose tissue. Awesome. So adipose tissue would go down, and I think you have heard from Chris Morrison about that, that also with the adipose tissue, there's leptin levels that go down. So that is the second part here. Changes are detected by receptors. So what is detected is leptin. That is the hormone that can be detected here. And we have leptin receptors, and they are basically mostly in the CNS. That's where it mainly acts. So from the adipose tissue, this signal of fasting gets propagated into the brain, and specifically leptin receptors are in the hypothalamus, in the brainstem. So this is basically the afferent pathway to the brain to let the body know, okay, something is up, we need to react. Then you have the central control center, and then there's going to be an output system. Does anyone have an idea what the brain's output systems are? Nicole, you have an idea?
The Vegas?
Vagus, awesome. What else? That would be the peripheral nervous system. What other mechanisms do we have to go from the brain back to the periphery? When you think about leptin. Yes, exactly, hypothalamic hormone, so the neuroendocrine axis. And what you also should not forget, there's also behavior that's going to be shaped within the brain. And so then it goes to the effector organs. So we change, with the fasting stimulus, we change hunger. We are much more hungry. We change food seeking. So we're going to be really motivated to go and get the food. And then if we have food, if we find some, we will actually increase our food intake, and energy expenditure would go down to really make sure that we can do much more with the energy we ingest than we typically would do. And so we are also going to focus a lot about energy expenditure today. Now, if you have done all this, you find food, you eat more, then the body fat stores are going to be replenished, and leptin levels will go up, and so we can go again into homeostasis. So another way to make it more simple, body weight homeostasis is basically the balance between food intake and energy expenditure. And so this one here, this image here, that is a study from Ruth Harris, who actually has been here in Baton Rouge and did research here. I think this study was performed here. They were basically forcing animals to reduce their body weight. And they were doing that by giving them, by feeding them with a tube, so the animal could not eat by itself. And one group of animals were fed 100% of food, what they regularly ate. And the other animals were reduced by 60%. They only got 40% of their typical food. And obviously the body weight goes down drastically. And if you then start to free feed them again, that they can choose whatever they want to feed, what you see here is that they increase their food intake, and particularly they increase their food intake above what the 100% fed animals were eating. And it's quite a while, so these are several days, like 20, this is 20, 30, so 10, 20 days that they eat more, and you see until they basically reach their body weight back again. And you can do the opposite. You can overfeed the animals here by 60%, and then when they are free feeding again, they will eat less than the 100% fat animals. So they are under eating just to make it to their body weight here. At the same time, so this is the food intake that's changing, there's also energy expenditure that is changing. So this is data where we fasted animals for only 6 hours, and within that short time the body perceives that it's fasting, and you see a hypometabolism that takes care of making sure that with that very little food that comes in, that you can maintain the body much longer, only until there's food again around. And so that's why how we kind of keep up with our body weight. So this is a typical growth curve that you see for children. And typically, wherever you are on this growth curve, you keep that stable pretty nicely, unless you have a defect in leptin signaling. And you see here, this is a child that doesn't have leptin. And you see that it really goes pretty much out of the schedule, and unfortunately you don't really see that very nicely here. So at 2 years, that child is about 20 grams. And And that is way, way up here. So you really get out of that typical growth curve if you don't have leptin. No, but then I told you a lot about regulating your body weight, but we all know that there is this problem in our society that the body weight is increasing. And so you see here the chart for the prevalence of obesity. That's a body mass index above 30. And from 1985 to 2014, you see a dramatic increase in the prevalence of obesity. So how does that fit with regulating your body weight? And we all know that because of that, we all feel there's a lot of pressure for strategies to fight obesity. I think everyone has seen in the stores there is 0% fat yogurt or sour cream. In fact, it's actually really hard to get something with full fat. There's sugar-free Oreo. It's even very crazy. There is calorie-free mayonnaise. You wonder why you would eat that, because it's not really food. And then there's a typical lifestyle intervention. Eat half of the typical meals that you have and exercise more. And typically that works very well. If we focus on it, we are motivated, we want to lose weight, it actually works. We will lose weight. However, over time, if you're not taking care and really make sure you force yourself to eat less, we always go back, and that is the big problem that we have in society, that it's very hard to comply with these things. I would like to make a case why that is. So let me skip this one. So this is an example of body weight regulation that very nicely shows how great our body gets it together. So these are hamsters here, and you see they look very different. And the Hungarian hamster is a seasonal animal, and during the summer it looks brown like this, and during the winter it has this nice white coat. So, but what is even more interesting is that in the summer coat, the animal weighs about 42 grams. And in the winter coat, the animal weighs about 27 grams here. That is a body weight drop of 30%. That is amazing. That would be awesome if we could reach that by treating obese humans. That would be really great. So this animal does it by itself just by changing the day length. So these are in a natural photo period, and where this animal lives, that means the days get much shorter during the winter time. And the experiment that they did was they wanted to find out what does the animal memorize. We all have heard about the body weight, that's the fitness, and there's a set bond that we try to come back to. What does the animal memorize? So if we start here during the summer and fast the animals down, so we force them to lose some body weight and then let them back free feeding, will they go back to this level, or will they go back to the level that they should have during their day lengths? And they know exactly from their day lengths where their body weight has to be. You can do that again, and they will go back exactly to that body weight where they have to be. So something, just by changing the day lengths, there's something in their body that tells them that's the body weight you need to defend. So that is amazing. If we go back to obesity, so these are rats that were on a high-fat diet, and they gain a lot of weight, and then they were food reduced or forced to eat less. That's like a diet that we have in humans for different times. So a few days, a few weeks, and here 16 weeks. And if you let them back to free feeding, they do exactly what these animals are doing. They go back to that body weight that they would have on that high-fat diet. So they memorize, if they're on a high-fat diet, that's the body weight that they have to have. So if you think about this one, it's really not that surprising. However, if you think about it, our general perception is that obese individuals don't regulate their body weight correctly. So there's somehow in our society there is this definition that people, if they're obese, they don't regulate their body weight. But the contrary is true, because they actually fiercely defend their high body weight. So that is, I think, very important to see, and it's really not that surprising because that's physiological adaptation. The only thing that's different is that the defended body weight has changed. And so my point is that if we could change or slide the body weight set point, we should expect that there's a voluntary adaptation of food intake and energy expenditure. So if we could leverage that, that would be really a great thing. And so in my lab, we're very interested to investigate the neural bases for changes in homeostatic set points because that would be really the ultimate goal to change body weight. Now, from this I would like to go to temperature homeostasis, and so if you talk about body temperature maintenance, it's really a balance between heat production and heat dissipation. And heat production is pretty much energy expenditure, and ambient temperature is changing energy expenditure in food intake, and that is nicely seen here. So we have here different ambient temperature, and the lower you go with the temperature, the higher the oxygen consumption goes, and oxygen consumption is basically energy expenditure. If you look at food intake, it's the same. The lower you go with the body temperature, with the ambient temperature, the more food you have to eat. So you have this regulation of energy expenditure and food intake, which is very similar to body weight regulation, where you also have this regulation of food intake and energy expenditure. So you wonder how different are these systems. And it's important to remember thermoregulation really has a nice dependency on ambient temperature. So the lower you go, the higher the energy expenditure, and then you come to a lower critical point, and that is basically where the body does not have to expend extra energy to maintain the body temperature above ambient temperature. And so there's a range where that is true, and then when you heat up further, the animal has to expend more energy to maintain to keep cool, basically. And so the basal metabolic rate is basically at this level where the animal is at rest, doesn't have to expend any more energy for heating up or cooling down. And if you are in this heating up phase, there you have to spend energy for vasodilation, you're painting, you're wetting the fur or seek shade to just try and keep cool. And then on the other part of that equation, if you need to generate more heat, you have to generate heat via the brown adipose tissue. That's something we're going to talk more about. And the other way to make sure you're not cooling out is to reduce the heat loss by vasoconstriction. And that can look, for example, like this. This is with a thermal camera. So this animal here, you don't see the tail. So the tail is very cool. The animal is vasoconstricting and keeps the heat within the body. And in this animal, you see there's vasodilation. So the animal tries to get rid of that extra heat in the body. Now, brown adipose tissue is really a heat-generating organ, and it has been typically studied in mice and hamsters. But it also has been, in 2009, it has been very clearly shown that there is brown fat in humans. And even though most humans at thermoneutral condition, which is when we wear all our clothing, they don't have a lot of brown fat, but it has been noticed by cancer patients that a lot of times when they're looking for tumors, the physicians were annoyed that there was something like brown fat that was looking like a tumor. So they used this deoxyglucose, radioactively labeled, to label metabolically active tissue, which should be the tumor, but also brown fat shows that. And you can actually induce brown fat by exposing humans to the cold, and you get a very nice expression there. And in obese individuals, it has been shown that there is reduced brown adipose tissue. However, be aware they are also insulin resistant, so it's quite unsure if that's really because they don't have as much brown fat, or if it's just not possible to get the glucose in. But very important to notice is brown adipose tissue is not only a specific depot, also white adipose tissue. So these are white adipocytes. They are big, have this big lipid vacuole. They can actually turn into brown adipocytes. So these are brown adipocytes, have these small lipid droplets in there. They have a lot of mitochondria, and that can be induced by the sympathetic innervation. And just to remind you all, the sympathetic nervous system is basically where the brain talks down to the spinal cord and then talks to the sympathetic chain ganglia that are right next to the spinal cord, and then innervate different organs. And the adipose tissue is specifically only sympathetically innervated. And if you expose an animal to cold, any adipose tissue can actually go through this browning process where you have more brown adipocytes. Even if it's pure white fat, you see very nicely here how the browning takes place, and it's different shades that you can get. So the capacity is actually quite high. Then also, how does brown adipose tissue generate heat? So this is the electron transport chain, and that pumps protons into the mitochondrial, between the mitochondrial membranes, and that is used to generate ATP, which is the unit of energy to do all the processes. And in brown adipose tissue, you have this protein, uncoupling protein 1, and that is circumventing this ATP production, and instead that energy gets released as heat. And that UCP1 is really important for brown fat function, and is demonstrated here where we have UCP1 knockout mice, so they don't express any UCP1. And in cold adapted animals, you have a lot of UCP1 expression, it's all active, and if you give norepinephrine, which is the currency of the sympathetic innervation, you get a huge induction of energy expenditure, whereas these UCP1 knockout mice do not respond to norepinephrine. So UCP1 is really required for brown fat heat generation. Now, brown adipose tissue has been also suggested to be important for body weight regulation, but it is actually very hard to nail that down, and people have tried to surgically remove brown fat depots, but that is in animals, obviously, and that only is a small part of all the brown fat that you have. They have tried to, and there's also a high rate of brown fat recovery, so it was all very unclear how good the conclusions were, because they never really found that it leads to obesity. So in 1997, Les Kozak, who also worked at the Pennington, he generated UCP1 knockout animals, and that was, everyone was waiting for this paper because people thought this would finally show that it's really important for obesity. But what he showed was that UCP1 knockout made these animals cold sensitive, but they were not obese. So everyone was very disappointed about it. And in follow-up studies, they showed if you put them on a high-fat diet just to boost the system and see maybe on a high-fat diet they would actually be obese, they actually found that these UCP1 knockout mice were leaner than the regular wild-type animals. And then it took till 2009 when Barbara Kinnan and Jan Nedergaard, when they used these UCP1 knockout mice, and they had the idea, well, maybe we have to use them at thermoneutral condition, at 30 degrees Celsius instead of at 20 degrees Celsius, like a regular room temperature, because at thermoneutral neutrality, you do not need your brown adipose tissue, you don't need UCP1, so there's no pressure on that system. And in that situation, they actually found that UCP1 mice were higher in their body weight. So this is on a control diet. If you put them on a high-fat diet, that is even more dramatic, that difference. So they proved, yes, UCP1 is actually important for body weight regulation. And this kind of demonstrates how important it is, how you conclude and how you look at your data. In that study here in 2003, they also put the animals at thermoneutrality, and if you look closely, they actually showed that the UCP1 mice started to gain more weight, and at the end of the study, they were no longer significantly different from the wild types, but they did not draw the right conclusion at that time. So they could have gotten that conclusion at that time, but they didn't. So, but now everyone believes that UCP1 is required for normal body weight regulation. And UCP1 knockout mice engage in alternative heat-generating mechanism. So in this condition, where they don't have UCP1, but they need to generate heat, they're going to be alternative circuitries. So that brings us to what actually is metabolic rate or energy expenditure. And so the energy that we take in is ingested, and assimilation brings the nutrients to other places that we need. It's not perfect, and some of that energy is going to be excreted as feces. And the rest that's uptaken is used to maintain our body metabolism. Then we have biosynthesis if an animal is growing. And we have work like exercise, walking, that's all comes together in energy expenditure. And any energy expenditure is basically substrate oxidation and the proton motive force that's in the mitochondrial membrane that we talked about. That is basically leading to ATP turnover and the basal metabolism. Not all of this coupling with ATP is perfect, and so we have some proton leak, and that also is basically together builds the obligatory metabolism. And then we do have active uncoupling, which is basically brown adipose tissue function, and that is the very organized proton leak that is happening. And there's also substrate cycling, which is happening when you don't have UCP1. And then the oxygen that is used for the electron transport chain and substrate oxidation, that is basically what we measure when we do metabolic rate measurements in humans or in animals. Now, so talking about different aspects of energy expenditure, so I talked to you already about that fasting induces a hypometabolic state. But then different ambient temperatures, they also change energy expenditure quite dramatically. So one question that we were asking is, what is if you are in jeopardy and both of these things are happening? You are in the cold where you want to increase your energy expenditure, and you're fasted and you want to decrease your energy expenditure. How does the animal handle that? How would a human handle that? And if you do that experiment, you actually see that you get the increase in energy expenditure, but you also have the same amount of hypometabolism, as if there are two parallel circuits that are happening. And as we just measure the overall energy expenditure, it's just a sum of all these different pathways. So if you would do the statistics on it, there's a very clear effect of fasting on metabolism to decrease it. There's a very clear effect on temperature, on increasing energy expenditure, but there's no interaction between fasting and ambient temperature. So these are the fasting-induced hypothermia is independent of ambient temperature, and there are likely different kinds of energy expenditure. And that brings us back to leptin as a homeostatic signal, and that's what we talked again before about. If we have leptin that we inject, we would reduce food intake as a negative impact on food intake. That leads to a decrease in fat mass, and that will feed back to reduce leptin levels. And so you know leptin is expressed in all adipose tissue. And if you fast animals here for 24 hours, you see that the leptin message in adipose tissue is dramatically reduced, but then upon refeeding, you can replenish this aspect. Now, how about energy expenditure? Energy expenditure is overall known to be increased by leptin, and that is true because if you record from the sympathetic innervation to brown adipose tissue, you can see that leptin increases the sympathetic tone. And increasing the sympathetic tone is basically stimulating UCP1, and you stimulate heat production. So brown fat temperature would go up, and that should lead to an increase in energy expenditure. But if you look at the overall body, the whole body, how does it propagate into it? So these are fat animals, fat mice, and you see the daily rhythm of metabolic rate. And here these arrows are leptin injections, and what you can see is nothing. Leptin does not change energy expenditure at all. But if you food restrict these animals, they will go into these hypometabolic states. And if you then treat with leptin, you see that leptin prevents this hypometabolic state. So even though leptin seems not to directly stimulate energy expenditure, it certainly prevents fasting-induced hypometabolism. And then to make it even more confusing, if you look at different, at leptin deficient animals, these are OB mice, you see that their body temperature is lower than in wild type mice. So yes, there is a regulatory problem with not having leptin, which may be very different than this situation here. And if you acutely expose them to the cold, you see this one here is actually the OB mouse, and it just cannot maintain its body temperature. That's why it's almost the same temperature as the surrounding. So, and in addition, if you cold expose animals, you will, similar to fasting, you will get a very rapid drop in leptin expression in the adipose tissue. So even though cold would increase energy expenditure and leptin would, and fasting would decrease energy expenditure, both of these stimuli would actually decrease leptin levels. So I'm not saying that we are understanding all this, but you might agree that at the very least we can say leptin has to do with this homeostatic regulation very clearly during this fat-fasted situation. And if we look at leptin receptors in the brain, those are a really good blueprint to learn about all these different effects of leptin, and specifically to figure out which of these neuronal populations are important for different aspects of energy expenditure. And so the question is, how can we connect the brain circuitries with the circuitries in the brown fat? And the pseudorabies virus is a virus that has been very well known to connect these peripheral circuitries. So it's a retrograde and transsynaptic tracer, and if you inject it here into the stomach, it will propagate into innervation, sympathetic and parasympathetic, and then label further upward circuitries all the way into the hypothalamus. And so that is something that my lab was doing. So we were injecting pseudorabies virus into the brown adipose tissue and wanted to see which neurons were basically were labeled with pseudorabies virus. And we did that in leptin receptor GFP mice. So in addition, we could also see where leptin receptors were. So in essence, where we see an overlap of here we from the brown adipose tissue, and you see that here in red, and leptin receptor. So here you see a lot of yellow in the DMH and in the preoptic area. So these are very specific sites in the brain. That would give us a good guidance that these neurons might be important for regulation of brown fat thermogenesis. And we were particularly excited about the preoptic area and the dorsomedial hypothalamus because there was already quite some literature known about thermoregulatory circuits, and that is from Sean Morrison's group and colleagues. And what they showed very nicely is that there's a circuit where you have the sensory part in the skin, and these neurons feed back to the hypothalamus, and this is here the preoptic area. And these neurons feed back to the DMH, and then you go all the way down to the raphe pallidus, and then back to the brown adipose tissue that is activated. So it's a really nice circuitry that they show that is activated by skin cooling, as well as by agents that induce fever. And so if you look at the neuronal code that is important, is one thing you have to keep in mind is the neuronal location. So where are your neurons located? So in the preoptic area or DMH. And then you need to know where these neurons connect to. And you see there are quite some lines here, how the connection is put together. And it's important to know, are these neurons stimulated or inhibited? So in this circuitry, cold is stimulating these sensory neurons that are connected to the skin. And you need to know what neurotransmitters are expressed. So it's different if you stimulate an inhibitory neuron or if you stimulate an excitatory neuron. And so in this circuitry, it has been shown that there's a glutamatergic input from the skin. And so glutamate is always stimulatory, and they stimulate a GABAergic population in the preoptic area, but they also are connected with another GABA neuron. So if you stimulate a GABAergic neuron, the net effect on the follow-up GABA neuron is basically that it disinhibits. And then it gives basically a possibility for this DMH neuron to fire and stimulate brown-fat thermogenesis. So that is what you are interested in if you look at neuronal circuitries. How do they fit together, and how does it make sense for the brain to regulate it, and how can you integrate that into different physiological situations like fasting and thermoregulation. Let me just skip this one. And the first area that we looked at was the dorsomedial hypothalamus, and what we also found is that these leptin receptor neurons are activated if we expose the animals to cold. And the neuronal activation is really nicely shown when you stain for the early response gene C-FOS. And what we saw here is that cold really quite robustly increased the neuronal activation in leptin receptor neurons within this particular population in the DMH. And then also if we treated with leptin per se, we also saw the same nice stimulation of these neurons. So one question that was really important is to look if this neuronal activation would actually translate into increased metabolism. So that is something you want to experimentally show that your neuronal circuit is really doing what you're hypothesizing. And during that time, it was actually two methods were put forward, optogenetics and pharmacogenetics. And it's really exciting because what you can do with these methods is to synthetically activate neurons. And you do that with help of a virus where you express this channelrhodopsin into a certain population of neurons. And channelrhodopsin is light sensitive. It's an ion channel that's integrated into that neuron. And when you shine light on it, you will open that pore and it will induce or excite the neurons. And what you can see here is basically a recording from one single neuron that has channelrhodopsin. And these arrows are basically light pulses, and every light pulse is giving an action potential, which translates into activating that neuron. The other method that is out there is very similar, but has some differences. So you express a designer receptor, which is a mutated muscarinic receptor, which no longer responds to acetylcholine as its endogenous ligand. It responds to CNO, and you have two versions of it. One is an inhibitory version, and the other one is an excitatory version. And so if you inject the animal with CNO or give CNO in the drinking water, you will see within that neuron, so that's again a single neuronal recording, you see that CNO increases the neuronal activation and it depolarizes that neuron. So it's a little bit different than this precise, every light flash gives one specific action potential. It's just overall, you just stimulate that neuron like crazy. And so another layer that you can put on it is that you really can express these systems in very specific neurons. And you do that by having a gene that you express, and it has these flux sites, the double flux sites. And the receptor that you want to express is basically in the antisense. So this cannot be expressed because the polymerase would jump off it. If you pair this with Cre recombinase, then you actually cut these flux sites, and it flips your gene of interest into the reading frame, and then you will get that expression. And by doing that, you can use a specific neural population. I think Dr. Morrison, he talked about AGRP neurons in the brain, so it's a very precise population in the arcuate nucleus and the hypothalamus. And if you have AGRP-Cre animals, you drive that expression, particularly in this arcuate nucleus. And if you give CNO, you see this nice induction of C-FOS in that area. So showing how nice you can target these. And so you can use whatever you want. Here, for example, leptin receptor specific, and then V-glut as a transporter for glutamate, or V-GAT as a transporter for GABA. So you can target GABAergic neurons, glutamatergic neurons, leptin receptor neurons. So it's a very nice and precise way how you can target these neuronal circuitries. These were two postdocs in my laboratory that worked on the DMH. And so the circuitry that we have is a preoptic area, innervates the DMH, and innervates the rough pallidus. And what we wanted to target is basically leptin receptor neurons within the DMH. So that's where we place the injection tube. And by using leptin receptor free animals, it would only express in leptin receptor neurons. Unfortunately, we don't really see that very well. So, but this is the expression pattern of leptin receptor neurons. We have here the DMH, and this is basically where we put cannulas in, and then you can infuse the virus that it will express, particularly in this dorsal part of the DMH, and that's how it would look like, the expression. So you have it coupled with the fluorescent protein, so you know exactly where your neurons are expressed at your target. And then if you look at the physiology, if you stimulate now specifically these neurons in the DMH that are leptin receptor neurons, what you can see is that there's an increase in energy expenditure. So here in black is basically a non-stimulated mouse that got saline, and these two experiments got CNO, and you get a dose-dependent increase in energy expenditure. You also get an increase in locomotor activity, which was unexpected. And then with the increase in energy expenditure, you also see an increase in body temperature. So here at injection site, there's no difference between the two animals. So the darker the color, the warmer it is. But then if you give CNO, you see this animal is 1 hour after receiving CNO, it's much warmer, the body. And this one is a vehicle-treated animal. So you can quantify it and you see nicely how they increase their body temperature. So you have increased energy expenditure and you have increased temperature. What do you think is going to happen with the body weight if you do that chronically?
No.
It should go down. Right. So that was exactly what we were trying to do. So we gave for 3 days CNO twice daily. And what you see is here the body weight is going down. And that is due to the fat mass particularly going down. And in this case, we actually did not see any effect on food intake, so it seems to be a very specific effect on energy expenditure. And it has the power only after 3 days to really modulate the body weight. You can also do an opposite experiment. Instead of stimulating these cells, we wanted to see if leptin really is doing something. So we took out leptin receptors from that area, and we used again a virus, and here we have leptin receptors that are excised or cut out with the AAV-Cre virus. And here we have exactly the opposite. Instead of increasing energy expenditure, taking away leptin receptors decreases energy expenditure, and that is increasing body fat. So this is also an experiment that we did to look at if these neurons that I showed you before that are brown fat related and show this nice co-expression of pseudorabies virus and leptin receptor, if these are GABA or glutamatergic. So this is a mouse model for glutamatergic neurons, so everything in green are glutamatergic neurons, and here everything in green are GABAergic neurons. And you see that the distribution pattern is very different. So here I have a lot of green above the DMH, and that's where the PRV mostly is. And here you have very little GABA in this area. And clearly all the glutamatergic neurons are also PRV neurons. And here the PRV neurons are not GABAergic neurons. So also confirming that these leptin receptor neurons are glutamatergic. So in a circuit, if you put that in a circuit together, we basically can confirm that these DMH leptin receptor neurons are glutamatergic, and stimulation of these DMH leptin receptor neurons promotes weight loss. It induces energy expenditure via brown fat and locomotor activity, brown fat activation and locomotor activity. It induces mild temperature changes. It has no effect on food intake. And so loss of leptin receptor has the opposite effect, basically showing that leptin is important on these, leptin action is directly important on these neurons. And it supports that leptin activates these neurons to stimulate energy expenditure. And we also show that these are mostly glutamatergic. So now if you want to connect the next step, the preoptic area to the DMH, so we did the same experiment basically by targeting the preoptic area to see if these leptin receptor neurons are also important for energy expenditure. And let me just brush through. And so what we saw here is that if we, so before I told you that these preoptic area neurons are GABAergic. So if we stimulate these ones, these neurons, and they would innervate the DMH, the prediction would be that the energy expenditure would be, if you stimulate a GABAergic input on these glutamatergic neurons, so you would think that the energy expenditure goes down because the glutamatergic neurons would be inhibited instead of stimulated. And so if we do that, if we stimulate these neurons, indeed we get this very dramatic decrease in energy expenditure. Or this is rectal temperature, and this is the energy expenditure. It really drops down dramatically. And so this is a video to show. So just to compare this effect with a torpor or hibernation, this is a hamster that's hibernating or in torpor. And you see they are very sluggish. They have their body temperature is super low. Here's the metabolic rate, here's the body temperature. And so if you start to handle them, they will try to increase their heart rate, but they cannot immediately wake up. So it takes them a while to get out of it. So this one looks very much like a torpor, right, with the metabolic rate here. But if you look at those animals, so this one here is saline. And this is a CNO-injected animal. You see, we try to handle them. You see, it tries to escape. But the CNO animal just sits around here. But you see, it does move. So that animal is very different than the hamster here. So it's not a full torpor. The energy expenditure effect is exactly this torpor behavior, but it's not with the consciousness. So this animal is still conscious, can run away, but the muscles are cold, right? So it cannot do the same rigorous escape than the other one. And also it's very dose dependently, so the more CNO we give, the more drop in VO2 and on oxygen consumption we get here. So overall this was very consistent that these would be GABAergic neurons, that if we stimulate these ones, we would basically inhibit these glutamatergic leptin receptor neurons and block the energy expenditure in this situation. So we wanted to confirm that these leptin receptor neurons are GABAergic neurons, and we had these VGLUT2, so these are stimulation of glutamatergic neurons, and we had VGAT neurons that are basically GABAergic neurons. So if we stimulate these glutamatergic neurons, we exactly recapitulated the effect of leptin receptor stimulation, while the GABAergic neurons did not do that at all. So that did not fit our equation. So these neurons were actually glutamatergic neurons, so there's no way that the glutamatergic neurons would have that effect on the DMH neurons. So we would have to come up with another circuitry. And so I would like to go back to mechanism of hypothalamic thermosensing. And so it has been shown by Berland's group that they are warm sensing neurons that change their firing rate with neuronal with ambient temperature. So the warmer the neurons, the ambient temperature is, the more they are firing. And then there are these insensitive neurons, which are the majority of neurons, and they are not bothered by any change in temperature. And then there are cold sensitive neurons that if you warm the ambient temperature up, they actually would decrease the firing rate. So we were asking the question if these neurons are indeed warm sensing neurons or cold sensing neurons. And what we did here to expose animals to cold room temperature or warm temperature. And then you see that only the warm temperature stimulated or activated these neurons. And in a sense, saying that these preoptic area leptin receptor neurons are warm sensing neurons. And so our argument was if these neurons are activated during cold, and you would activate them in addition with the synthetic activation, you would think the effect would be less because they're already on. And if you're on cold temperature where they would be switched off, and then you stimulate, you have a larger range to stimulate them. And that's exactly what we saw. So if we change temperatures to thermal neutrality, we see that even though there's still an effect on energy expenditure, it's very small. And if we go to 10 degrees Celsius, see that the change in energy expenditure or drop in energy expenditure is much larger at 10 degrees Celsius. So in essence, we have a temperature-dependent effect of this activation of leptin receptor neurons. And in a sense, that means that these preoptic area leptin receptor neurons, they drive temperature-dependent energy expenditure adaptation in food, and it was not quite clear if that also propagates into the adaptation to food intake. As I told you, changing temperature would regulate energy expenditure and food intake. So energy expenditure, we show these leptin receptor neurons do drive the effect on temperature-dependent energy expenditure. And also, very important, the DMH neurons do not do the same thing. So even though these DMH leptin receptor neurons drive energy expenditure, it's not temperature dependent, showing this segregation into two different kinds of energy expenditure. So to look at food intake, we did an experiment where we also wanted to see how the body weight is regulated. So what would you expect if the energy expenditure is low and you get chronic CNO? What would you think what would happen with body weight?
Increase.
It should increase. But there was not what we were observing. Despite this super low energy expenditure, they decreased their body weight over a chronic activation of these neurons. And that is because the food intake also dropped down quite significantly. So the net effect is a decrease in energy expenditure. So it's important to get the whole equation. You cannot just look at energy expenditure and do your predictions. You need the food intake as well. And also the food intake was very nicely regulated dose dependently. So the more CNO we gave, the more decrease in food intake we got. So overall, this showed us that the original view was actually no longer sustainable, at least for the leptin receptor circuitry. So even though leptin receptor neurons are robustly, they robustly decrease energy expenditure and body temperature, this was not due to GABAergic activation, it was due to glutamatergic activation. And so we have to conclude that these neurons cannot directly innervate the DMH neurons because in essence that would actually stimulate energy expenditure. And that is still something that needs to be figured out. Now after we published these data, there was a very nice experiment done by Zachary McKnight, and he's actually coming next week and gives a presentation about his data. And they used a different approach to look at warm activated neurons. So instead of doing brown fat related neurons that they were looking for, they specifically looked for warm activated neurons within the preoptic area. And because warm is known to stimulate these neurons, and that would increase CFOS, but it would also increase this ribosomal phosphorylated protein, phospho-S6. They use that to tag ribosomes. So if you, with an antibody, pull down these phospho-S6 ribosomes, you will get basically all the RNA that's associated with this neuronal stimulus. And so they did an RNA sequencing with that, and they found certain marker genes, so some marker genes for neuronal activation, FOS2B and FOSL2, and BDNF and PACAP were also two proteins that they found. And so you see they were very similar expressed, They did not find leptin receptor in this experiment. And when they did the same experiment that we did and stimulated these neurons in the preoptic area, they also got a decrease in energy expenditure. But you might notice that whereas our body temperature dropped down 6 to 8 degrees Celsius, this is a fairly mild drop that more made us relate to the DMH effect. So this is about 1 to 2 degrees Celsius at that drop in body temperature. And also what they found is that these animals do have a vasodilation, so they try to get rid of heat, so they cool down by losing weight via the tail. What they also found in this study is if you specifically stimulate projections from the preoptic area PACA, BDNF neurons to the DMH, so it's a very select subset of these neurons, they also can basically show this decrease in brown fat temperature. And you see again, this is a very mild effect on energy expenditure. So these warm activated preoptic area neurons are also GABAergic, that's what they showed, instead of the glutamatergic leptin receptor neurons. They have this mild decrease in body temperature. And they also showed that these are related to temperature-seeking behavior. So these neurons seem to be the one that are GABAergic and innervate the DMH, and they could innervate the DMH leptin receptor neurons to then, if you stimulate GABA, you would inhibit this axis and would decrease energy expenditure. And if cold would basically decrease these neurons, the neural activity, then you would allow brown fat activity. And in contrast to that, there was another paper that came out that also looked for warm activated preoptic area neurons, but they did that by electrophysiology, and they identified neurons that express TRPM2, which is a thermosensitive ion channel. And these neurons are actually glutamatergic neurons. And when they activated these neurons, they actually got the same really dramatic decrease in body temperature that we got with our glutamatergic leptin receptor neurons. So this is about 6 to 8 degrees drop in body temperature. And these neurons were important for fever inhibition. And they also showed that these neurons act via the PVH. So in essence, we can say there are these different circuitries that was not known before. So that is really something that the field knows right now much better, that there seems to be this original circuitry that had been demonstrated by Sean Morrison, where you inhibit the DMH, and cold would basically disinhibit this DMH circuitry to allow energy expenditure. And then there are these glutamatergic leptin receptor or TRPM2 neurons that were not known before. So that is actually a new circuitry that's brought up here that seems to act via the VMH and is important to regulate fever, torpor, and these deep changes in energy expenditure. And so one question that remains is how this relates to these different situations of fasting-induced hypometabolism versus ambient temperature-regulated energy expenditure. And I think this is particularly interesting to understand how obesity would relate to these metabolic systems. So far, it's really unknown if obesity is something that is only regulating the hypometabolism of fat, fasting, and refeeding, or there might be also defects in thermoregulatory circuitries. And with that, I would love to get some questions if you have some. Thank you.
What are your next steps for project working?
So we're particularly interested to see the fat fasting effect versus temperature-dependent effect. So we already identified the preoptic area as being very ambient temperature-dependent, and the DMH is not. So right now we are looking into this fat fasted situation versus DMH and preoptic area. And we have some data that leptin seems to be important for this fat fasting effect within the preoptic area. And so that is something that we try to piece out. And the other thing would also be to translate it to humans or to animal models of obesity and see if they particularly have a defect in regulating energy expenditure or food intake to ambient temperature versus this fat-fasted refeeding situation.
So do you think that in humans is also related to the demographic area where people live?
It's a very good question. I think some people have tried to do that. I don't think it has been very conclusive, but there are certainly differences in probably fat distribution. For Eskimos, for example, they actually do have more brown adipose tissue, so you do get an induction if you live in cold areas. But I don't think people have really tried to leverage that to treat obesity at this point.
I have a naive question. With this 6 to 8 degrees drop, how is that related to energy, especially in terms of kilocalories or calories? Because I just can't connect the energy expenditure in terms of calories directly to the temperature change. It's just association.
I mean, I think it depends a little bit. So for example, the DMH has this mild effect, and I mean, somehow I'm reminded of this fasting response because fasting has this mild hypometabolism. And in those nodes, you also don't have a change in food intake, whereas with the preoptic area, you have this dramatic decrease. You also have the decrease in food intake. So kilocalorie-wise, it's very hard to tell. And I mean, we just, by coincidence, we really noticed that we were expecting them to gain weight and they decrease weight, and that's why we looked more detailed into food intake. So absolutely, you have to put the whole equation together. I mean, I cannot tell you exactly what degree Celsius would come up with what kilocalories that you would save, basically.
So in that condition, experimentally, can you put the animals, like tube feed them with the same amount of calories regardless whether the temperature drops, so that way you have, their intake doesn't change, well, you're basically manipulating and giving them the same intake.
Right, I mean, so for the animals that don't eat, we would have to tube feed them probably to force feed, or we would have to have some other animals that we peer feed to a low amount. So that's going to be difficult. What we are trying to do to associate it to defects in this response to fat fasting. So for example, if we modulate these neural circuitries and then put them in the fasting situation, can we block or can we enhance the fasting-induced hypometabolism? So preferably we would like to block that, basically, that you no longer get the hypometabolism. And then you could look how much more or less would they, so you would think if you block that, they would enhance their body weight. And theoretically, if you have a human who is on a diet, I mean, this hypometabolism has a severe effect on the amount of calories that you can take to maintain your body weight. So theoretically, we would be happy if we could block that response to make a diet more efficient.
So I have a question. Human infants are born with brown adipose tissue, so I think of it as preparing them for outside the womb body temperature. Can you comment on that or add something?
Yeah, so definitely all or most mammals, when they are born, they have a lot of brown adipose tissue. That was the original, the classic view that brown fat is basically to get them into surviving when you carry a baby over so cold room temperature. However, nowadays it's also known that brown fat is existent in adults. So that is something that really is basically under investigation, and Leanne Redmond, she's basically very interested to figure that out. There seems to be some association of also the mom's energy expenditure during pregnancy and the outcome of the babies to have obesity or not. And so the brown fat would be basically something that's very interesting to look at if that has any effect on the metabolism overall. But there's really nothing as hardcore data that we could say that's important. But I think overall it shows thermal regulation and body weight regulation is so close tied in. I think we need to think about it much more. And then all the human data that we have is energy expenditure. That is a mix, a sum of all these different energy expenditure pathways that are activated or inhibited. And we need to keep that in mind. And the first step is to understand how it actually regulates physiology. And then we can ask how that makes sense then for treatment of obesity, or how we can look at babies, how that is affecting them.
Should be interesting. I'm just wondering if say overweight women have infants who have more or less brown fat. We really, do we know anything about that? I'm asking because I don't know.
I mean, as I showed, so there is data that there's less brown fat in obese humans, but the problem is with the method of this PET scan, it is basically uptake of glucose. And as you typically also are insulin resistant, this could also just reflect that the brown fat is resistant to insulin, and that's why they don't take it up. It does not necessarily has to reflect that there's more or less brown fat. That is something that needs to be solved on a technical basis, basically. And people are trying to find different markers that are not so dependent on insulin to measure brown adipose tissue. And that's going to be key.
Okay. Because
we're not there yet.
With an infant, if you could see what happened after birth to the brown fat.
Right.
Might
And I think Dr. Carmichael, he's working intensely on that to try if there are any other markers they can use to non-invasively measure that, because no one wants to have a radioactive tracer in a child, in a baby. So there are certain difficulties that make it very hard to go on with these experiments. But, we have to build the basics in animal studies, obviously.
So I guess the other thing is, you mentioned that white fat can be converted to brown fat. It could go both. Can brown fat, does it go both ways? Is it the arrow go?
Well, so it's not so much that, if you have the brown fat, that stays brown fat, but it will look more white fat-like if you adapt to thermal neutrality. But you will always keep a core of UCP1, more mitochondria, so it does not entirely go back. But all the white adipose tissue that can be brown and cold, if you put the animals or humans back to thermal neutrality, it would completely white out and would not have any brown fat.
So it takes those cells out, whereas the others just tend to expand, or the brown fat expand or decrease in size?
So they are pre-adiposized in the depot, and they seem to be able to change into brown fat cells. And there's a whole other research in there. So apparently it's not as simple brown fat and white fat. So the pure brown fat cells are actually muscle cells in essence, whereas the brown fat cells in white adipose tissue, they are really from white fat precursor cells and change in brown fat cells. And there also seem to be cardiomyocytes that can also propagate into different kinds of brown fat cells. So there are these different genetic markers right now, and people are very interested to look into it, who is doing what. But overall, humans are thought to have mostly the white fat adipocytes and no longer the skeletal muscle adipocytes that go into brown fat.
It's very interesting.
It's complicated.
Correct, yes, it is complicated.
Are there human conditions or diseases where people have defects in thermal regulation that have genetic underpinnings that we know of?
For the brown fat,
Not necessarily brown fat, but just people who can't regulate their body temperature very well.
I'm sure that, they are.
Sclerosis, MS, that's one.
That's true, yes, that's true. And also all these neurodegenerative diseases, it is very well known that specifically in the late stage, they do waste a lot. So they really lose weight very rapidly. And there seems to be an effect on thermal regulation Whether that is because the brown fat is kind of crazily starting to be induced, that is unknown, but there seems to be differences if you're exposed to different temperatures, that is true. But it's not very well studied overall, so that's why I am also more and more interested to bring that in, so we don't really think a lot about thermoregulation with diseases.
Other questions, comments?
All right.
We'll have to wait for the next chapter. It's all, it would be very, very interesting as you talk to the see what
Yes.
that they're trying to get at.
So we try right now, we work together on a project with HUEC China as well, and we're trying to get a basis on for that is basically fitting for thermal regulation, but would also hopefully fit for these humans that have higher or lower energy expenditure when they're pregnant. And then to see how that changes the outcome for the offspring, basically, if they are more less obese or more prone to be obese or not. But there's very little that is known, and to put it into the human level, it's difficult.
Right, and I kind of think that brown fat.
I don't know, I think she was design. I think she was probably
chemistry insurance and they come out of the womb and they're in, they're in this environment, so that.
Right, so that's overall
Some kind of
the same.
self-regulation body.
Right, but pigs, for example, don't have any brown fat. And I know they are more sensitive to cold exposure overall.
Okay.
But it's not that we do have some other mechanisms that can be used as well, as was with the UCP1 knockout mice. They have this substrate cycling, so they can do some additional stuff.
And actually, the pig that if you're looking for an animal model that is closest to the human in terms of development, is, I
The
mean,
pig
that's
is really good,
but-
yes,
yeah,
that's true.
in terms of.
See, China just recently generated pigs with UCP1 or brown fat expression, so they are actually leaner. So you can make them into fit pigs.
Interesting, isn't it?
Yeah. So people are very interested in the brown fat overall, so I think it's gonna come that people have thermoregulation more on their radar.
Yeah.
And I think it might be used to, classify
it's off.
obesity into